Hi, my name is Julian Paganini. I'm a PhD student at the IMMEMXIII Utrecht, and today I'm going to show you a new method to reconstruct plasmids that contain antibiotic resistance genes on E. coli. The first thing we need to understand is that plasmids are mobile genetic elements that frequently carry genes that provide resistance to antibiotics, and these are particularly dangerous because they can be transferred across bacteria of the same species and of different species, and they can, of course, spread this antibiotic resistance. Another thing we need to understand is that the most common sequencing technology out there is Illumina short reads. And this is a great technology, but it poses a problem. After assembling these short reads, we will obtain a series of longer fragments of DNA sequence that are called contigs, but these contigs will be completely mingled and tangled with each other in an assembly graph. And out of the box, we don't know if these contigs derive from a plasmid, from multiple plasmids, or from the chromosome. Okay, so the method that we developed consists of two steps. In the first step, we will label each of the nodes in the assembly graph as either a plasmid or a chromosome. To do this, we developed a new tool which is called Plasmid-EC, and Plasmid-EC is basically an ensemble classifier. What it does is it takes every node in the assembly graph and it runs 3 different binary classification tools, and out of the majority votes it provides a final output for the classification. So each node is labeled as a plasmid or as a chromosome. In the second step, we will bin the nodes that were classified as a plasmid into individual plasmid predictions, as you can see here. To do this, we use G+. G+ is a tool that creates random walks through the assembly graph and connects neighboring plasmid contigs that have a similar sequencing coverage. So obviously we wanted to test how our method worked, and to do that, we downloaded 200 complete E. coli genomes from public databases, and what we did was compare the performance of our tool versus other plasmid reconstruction tools that are already available in the web. These other 2 tools are called IMMEMXIII and plasmidspace. So what we basically did was first test the capacity of the tool to reconstruct the plasmids. And the way we did that was basically comparing the plasmid predictions against the true plasmids that we got from the reference databases. And basically the way that we compared them was using 3 different metrics. One of the metrics is called recall, and recall basically allows to answer the question, how complete is the plasmid prediction? As you can see here in this picture, if our plasmid prediction is 30 kilobases and the true plasmid is 60 kilobases, we will assign a recall of 0.5. The other metric that we used was precision, and precision allows to answer the following question: is the prediction contaminated with other plasmids? So it's a measure of how pure is the prediction. So for instance, in this case, if our prediction contains 80 kb of plasmid A, but 20 kb from plasmid B, precision will be 0.8. And then we have a global metric, which is the F1 score, which encompasses both recall and precision. So we evaluated the predictions made by the 3 tools using these metrics, and what we found is that our method, which is called G+ Plasmid-EC in this plot, presents a much higher F1 score than the other 2 tools. The difference between the other tools, it depends on the metrics that we are evaluating. So, for instance, our method and IMMEMXIII present a similar precision, but the precision of plasmidspace is much lower, and our method presents a higher recall than IMMEMXIII, but a similar recall to plasmidspace, which actually means that our method is taking the best from both worlds. And this particular difference we can see when we are reconstructing plasmids that contain antibiotic resistance genes, which are the most difficult plasmids to reconstruct. The other thing that we evaluated was the capacity of the tool to assign the antibiotic resistance genes itself to the correct plasmid backbone. And you can see these on this plot. Here we have our method, MOPSUD, and plasmid spades. And here in orange, we can see the amount of antibiotic resistance genes that were assigned to the correct plasmid backbone. And this was evaluated by the antibiotic resistance genes that were assigned to a plasmid reconstruction that had an F1 score higher than 0.75. And as you can see, our method performed better than MOPSUD and even better than plasmid spades. So as a main conclusion of our study, we can say that our method, which combines G+ with plasmid EC, is the best method to reconstruct plasmids that contain antibiotic resistance genes on E. coli. And we saw that because it presented the highest F1 score and it was able to correctly assign most antibiotic resistance genes to the correct plasmid backbone. Our method also shows the importance of using assembly grafts to reconstruct plasmids.